Good morning and welcome to the Minnesota Stormwater Research Spotlights, which is part of our Minnesota Stormwater Seminar Series. Our Minnesota Stormwater Research Highlights are highlighting research projects that we've completed through the Minnesota Stormwater Research Council. The Minnesota Stormwater Seminar Series, which operates every other month, brings in nationally recognized experts of advanced stormwater intervention and knowledge, and share that with all of us attending these events. As a reminder, all of our upcoming schedule and our past recordings are available on the website. I will show that multiple times, and I will put the website in the chat box. So many of you have participated in our past seminars, featuring a large variety of experts from across our country. And so we will continue our Stormwater Seminar Series in the upcoming months. And so I'd like to highlight that schedule in just a moment. First of all, I should begin with a welcome. My name is John Bellotta, and I'm with the Minnesota Stormwater Research Program here at the Water Resources Center at the University of Minnesota. I want to thank the Clean Water Land and Legacy Amendment and the Minnesota Stormwater Research Council for providing financial support that makes the seminar series possible. I also like to thank my co-hosts, Dr. John Gulliver and Dr. Andy Erickson, and all the team over at St. Anthony Falls Laboratory that partner with us to bring this successful seminar series to you over the last couple of years. Before we launch into the seminar series, I do want to highlight a few things. Our new 2021 highlight of the Water Research Program is available online. So I would encourage you to take a look at it. It's a quick read, a 4-page summary of all the research efforts funded by the Minnesota Stormwater Research Council and our work currently going on. I also want to do a little plug for our funding campaign to increase our research pool for future years. So we have a very diversified approach to funding stormwater research here in Minnesota. One of those pools comes from campaigns of contributions from cities, watersheds, and private businesses. And this year's goal is for us to pool together $175,000 to help us leverage against the Clean Water Legacy funds and other funds that we get from USGS and other sources here at the Water Resources Center. So please join us in helping us obtain our goal so we can complete future stormwater research for our program. With that, I'd like to also alert you that our Stormwater Seminar Series will continue for the remainder of the year. Coming up here October 21st, we will feature Dr. Robert Pitt, continuing our effort to look at biofilter media. In November, which is November 17th, I'll alert you that's a Wednesday, a change from our typical Thursday. We are going to feature Greg Wilson from Barr Engineering, and he will present to us his conclusions on our stormwater pond lime treatment study. Please join us for that. And we'll conclude 2021 with a feature presentation from Michael Dietz from University of Connecticut. So please join us for all of those. And we are pleased to announce that we will continue the Stormwater Seminar Series in 2022. We're still in the planning stages of that. We always welcome speaker suggestions. So if you have those, please send them our way. A reminder this morning as we go into our seminar, please use the Q&A function which is located in your panel, not the chat function, the Q&A, to ask questions to our presenter this morning. And a reminder that using that feature, you can also upvote and give thumbs up to other people's questions. So I'll know which ones people have the most interest in hearing answers from our presenter. So with that, I am going to introduce our speaker and our topic today. We are pleased to have Dr. Andy Erickson, my co-host in the seminar series, present today. But Andy is also, as you know, an expert and esteemed researcher, and he's been working on a couple projects funded through the Minnesota Stormwater Research Council, And one of those is on biofiltration media optimization. And this work, again, funded by the Stormwater Research Council, has a number of phases. I'm sure all of you read the description, which brought many of you here today. I see we're up to 81 participants. We're expecting about 150. And so I'd just like to welcome Andy Erickson today. Of course, he is a research associate at St. Anthony Falls Laboratory and here at the University of Minnesota. His research looks at understanding water quality in urban and agricultural watersheds and looking at other stormwater treatment practices. Some of you may know him on his work, looking at iron enhanced sand filters, and he's contributed a lot to our field. And so thank you, Andy, for joining us today, and I'll let you take it away from here.
Very good. Thanks, John. Welcome, everyone. As John said, my name is Andy Erickson. It's my honor and my pleasure to be here with you today to talk about our research on biofiltration media optimization. We will talk about some of the phase 1 results. We'll get into the phase 2 preliminary results, as well as finish up with some future work. Now, this project has a rather large project team. So we have a core at St. Anthony Falls Laboratory, including myself and my co-lead, Jess Kozarek. We also have Katie Kramarczyk and Laura Lewis on site doing a lot of the work. Laura graduated actually in May and did a lot of work on this project, but now is on to bigger and better things. We're very thankful for all the work that Laura put into this. We also have Barbara Heitkamp and John Chapman from NEU. We're helping with outreach, at least through phase 1. John Chapman is still on through phase 2. Barbara Heitkamp has also moved on to another position. We're very excited to see that change. We're excited for her to have that. But we also have a number of stormwater professionals on this project. So Mike Eisensee, Mike Trojan, David Fairbairn, and Aaron Anderson-Wentz, we're all project team members on phase 1. And we're very thankful for the input that they provided. Our phase 2 team has slimmed down a little bit, but we still have a very strong technical advisory team, which I'll talk about at the very end of this presentation. But before we go any further, I want to ask the question that I'm sure many of you are asking. What is biofiltration, right? We use a lot of these fancy terms in stormwater management, and some mean the same thing, some mean different things. Some things mean different things to different people. So what I want to do is kind of give a context and a definition right up front. And biofiltration are shallow depressions, similar to rain gardens, similar to bioretention practices that capture polluted stormwater from our landscapes. But what's different about a biofiltration practice is that there is a engineered or designed, specifically chosen media through which that stormwater flows vertically downward. And in ideal situations, that media treats that stormwater before the water is collected through a gravel subbase and perforated outflow pipe, underdrain pipe. So the water then goes back to the storm sewer system, ideally in a treated state. And the mechanisms here are filtration at the surface because it is porous media flow. We have vegetation, that's what makes it a bio-based system. But then also we have soil microbiology going on in the media, right? So the combination of the vegetation and the soil microbes are also breaking down soluble pollutants. The challenges we've seen with these biofiltration practices is that we've seen phosphate release. We have polluted stormwater coming in, water going through that media, but then it's collected by an underdrain. It goes back into our storm sewer and often back to surface waters. So if phosphorus is being increased, if there's more phosphorus coming out of this system than what goes in, that's going to have a big impact on our water quality. And in freshwater temperate zones, especially Minnesota and a lot of the Midwest states and other parts of the country and around the world, in these freshwater systems, phosphorus is often what is limiting our algal blooms. So if we have too much phosphorus, then we have too much algae, usually. What other issues we see in these biofiltration practices include slow filtration rates. We get ponding in these basins often for too long. And in those cases, and in other cases, even where we don't have ponding, we can have poor vegetation growth. We don't have a healthy vegetative or soil microbiology state or community. And then that limits our treatment in these systems. So how do we overcome that when we're talking about a biofiltration practice? Well, as engineers and designers, we're often trying to select a media that gives us an appropriate filtration rate that supports the vegetation and the microbial community, and possibly could also remove other pollutants. So the base of that media is often ASTM C33 concrete sand. It's the clean washed sand. But then we add usually organic matter, and it's typically a leaf-based compost. Many state stormwater manuals, in the United States at least, limit that organic material to leaf-based compost. Although some states do allow food residue or other organic material to be used. I've also seen in recent years modifications to this design, this media mix. Peat, for example, very prevalent in northern Minnesota. We have a very abundant supply, but also others have been trying to put in iron into these mixes. If any of you are familiar with some of the work that we've done here at St. Anthony Falls Laboratory in the University of Minnesota, we developed an iron-enhanced sand filter specifically for stormwater treatment and capturing soluble phosphorus. We've seen that being incorporated into these rain garden designs. And then, of course, there are always other things that we can mix into this media to try to achieve these goals that we have for biofiltration. Before I get into our research, I do want to highlight the benefits of compost. So compost and organic matter obviously supports that vegetative growth. And that provides aesthetics, it provides pollinators or native grasses. It actually benefits a hydrologic cycle through evapotranspiration. We also support the microbial communities within the soil, in the rhizosphere, in the area around the roots of the vegetation that can break down nitrogen species such as nitrate. It can break down polycyclic aromatic hydrocarbons or PAHs. It can also break down carbon-based compounds. So pollutants that are coming in with stormwater and getting into this media, those microbial communities can break that down, convert it to gases, typically nitrogen gas and CO2. But that organic material also has moisture holding capacity, which further supports the vegetation and the microbial communities. Compost itself and organic matter can absorb metals out of the runoff at relatively high rates and for a long period of time. And compost is a reused material, right? We are composting a lot of our natural organic material, and that is sustainable and reused material. So there's a lot of very positive benefits to compost. And I don't want this presentation to seem like we're pointing the finger at compost and saying compost is a bad thing. That's not true. There's a lot of benefit. We need to find a balance between all the benefits compost and organic matter provides with some of the detriments that can happen in these biofiltration systems. And that's really what our research set out to do. We wanted to develop better biofiltration practices by identifying and using local sources of media. We don't want to try to find a media that comes from a far off place that costs a lot of money to get to Minnesota or in other parts of the country. We want to be able to identify readily available local sources and sustainable sources of media. We also looked at in phase 1, developing simple tests and metrics that could help us identify organic material or other media that might leach phosphorus before we actually put it in the ground. And then finally, we wanted to take all this information and develop optimal design specifications, specifically with 3 objectives in mind. The first, we wanted to have an acceptable filtration rate, because if the water can't be filtered by the practice quickly, we're not going to be able to treat a lot of water, or we're going to have to make that practice really, really big. And if it treats it too quickly, then we can't get the benefits of that media filtration and that absorption capacity and other chemical processes and biological processes that can happen in that media. So we need to find the Goldilocks of the filtration rate, and we want it to be just right. But our second objective is we want to support the plant growth and the microbial communities. Again, if we don't have plants, we don't have microbes in the soil, it's no longer a biologically based system, and we lose out on all of those benefits. And then finally, as I mentioned, we've seen some of these biofiltration practices releasing phosphorus, exporting more phosphorus out than what is coming in. And that is something we want to eliminate or at least mitigate as best as we can. So these were our primary objectives when we set out in phase 1. I will talk about phase 2 as well a little later in the presentation. But to give you a highlight of the goals for phase 2 as we move into that, we wanted to evaluate multi-year performance, Phase 1 paid for the first year of study. With the phase 2, we actually are going to get 4 years, 4 summers worth of rainfall data through our experiments, which is very exciting. We also propose to put in new mixes, which I'll be able to give you some highlights from our preliminary data of that today. So I'm sure many of you are excited to see that. We also are going to investigate phosphate release in response to road salt. So in cold climates, in northern climates, we use road salt in the winter to mitigate our ice and keep our roadways safe. But that can have some negative effects on our stormwater control measures and our stormwater treatment practices. We wanted to know, in a biofiltration-based system where the water is collected by an underdrain and exported back out to our storm sewer systems, does road salt mobilize any phosphorus that was previously captured? And if we can measure it within our statistical uncertainty, does it affect our vegetation growth? We also wanted to look at low organic content mixes and find out, can we get seeds to germinate if we reduce down that organic content to a certain level? Is there kind of a lower threshold below which we're just going to really struggle to get anything to grow initially? So we wanted to find out, what is that low end? A lot of these items and these goals that we have for phase 2 actually came from our technical advisory team, which is made up of stormwater professionals and industry experts. And so we're very thankful for that group to give us input into these projects so that we can guide the research to get answers that stormwater professionals like yourselves are interested in. So how did we complete this work? How did we obtain our objectives? We had several different types of tests. In phase 1, we conducted some batch tests, very simple mixing tests in jars to see if we could find some very simple tests and metrics that could help us predict release of phosphorus. To do this, we took a number of compost and organic matter mixes from local sources. We put them into small jars like you can see in the picture here, and we mixed them for a period of time just with water to measure how much phosphorus leached out of that organic material. And then we also collected samples of the organic matter and sent it off to test for sulfida, brave phosphorus, also phosphorus, and other metrics, which you're going to see on the next slide, to see if there's a correlation between how much phosphorus releases into the water and some of these simple tests of the organic material itself, with the thought that it could be tested before it's installed or even before it's delivered to tell us whether or not it should or may not release phosphate. So as I mentioned, we looked at a number of different metrics and analytical techniques. All the details are in the full report, which I'll link at the end of the presentation. But as you can kind of see through these graphs, I'm not going to explain all these graphs, but we tested a number of different metrics and tools. We didn't find any strong correlations. There was a lot of variability from the different organic materials that we got from 5 different local sites. And they were a mix of either food residue compost, yard waste compost, or a blend of composts. And all of them exhibited a high amount of variability in the phosphate that was released and variability in all of these different metrics. So we really couldn't find something that could give us a good predictive tool on this material, which is unfortunate. We'd hoped to find something simple, but what it tells me is that the variability is very large. So you never know what you're going to get, and you need to be very careful. And there needs to be more work here. One thing that could be really beneficial is perhaps doing a survey, doing a site survey where we collect samples from a much broader, instead of just 5 sites, let's collect samples from a very broad spectrum of sites all across the state or even the region, and do similar analysis and see better statistical representation when we have a much larger sample size. But from this, we really couldn't correlate or make any good, strong predictions. Another set of tests that we're doing, and this is actually happens to be part of phase 2, and this is coming up in future work, which we'll see at the end. We're also going to do seed germination tests, as I just mentioned a couple slides ago. These are small greenhouse type experiments, which you can see in the photo here. We want to look at low organic content mixes, possibly also looking at amendments, depending on the first set of experiments go. And we also are looking at a broader spectrum of species. We actually, again, we turn to our technical advisory team and our stormwater and industry experts to tell us what vegetation species represent the very large palette of vegetation that can be installed in biofiltration practices. And we narrowed it down to a list of 10. And so we are going to look at that. This is our current plan. So along the horizontal side here in the blue box, if you can see my cursor, these are the mixes we're going to look at. And it's anywhere from 0% compost with 100% sand all the way up to 20% compost and 80% sand. And then 3% compost, 5% compost, and 10% compost in between. The numbers in the boxes here are number of replicates. Because we're doing a germination study and it's vegetation, we need to have a high number of replicates to give us any kind of statistical confidence in this data. And then on the left side here in the vertical boxes in the green bars, these are our 10 current mixes or seeds that we're planning to put into these mixes. And so again, we have a pretty broad spectrum. We have fescues, we have cold season grasses, warm season grasses, a number of different things to represent the large palette of seeds that we could use in bioretention and biofiltration. So this is our current plan, and this will come up in a later stage, which I'll talk about at the very end of the presentation. But the bulk of this presentation is going to talk about our mesocosm experiments. And these experiments span both phase 1 and phase 2. Mesocosm literally translates to medium world, and it represents an experiment that's between the very small scale, the batch tests, the simple column tests, and a full scale or even a larger pilot scale. In our case, we are doing these outdoor because it is a biological system. We want it to grow naturally, be in the environmental conditions. They are trash can size, so they're roughly about 3 feet tall and about 1 foot in diameter. But we are dosing them with synthetic runoff so that we can control those variables. With these experiments, we can measure the filtration rate, we can measure the vegetation growth, and we can measure the phosphate capture or release. In the first 2 years, we had this set of 10 different mixes. We looked at 100% clean washed sand, which is our baseline. It's a non-organic mix, it's just sand. We also looked at 2 different types of compost. We had food residue compost and leaf-based compost in ratios of 10% and 20%. Now, if the percentage is not listed, that means the remainder is sand. So if I have 10% food residue compost, that means 90% by volume is sand, okay? And that applies to all of these 10 bullets. We also looked at, could we replace the compost with a different organic material like sphagnum peat or reed sedge peat? So we have 2 mixes where it's 20% sphagnum peat and 80% sand, and then 20% reed sedge peat and 80% sand. But then we also said, well, what about amendments? Could we mix something else in here to maybe offset or mitigate the phosphate that might be released from the organic material? So we put in 15% biochar, which is a hot topic these days, with 20% leaf compost, and then the remainder being sand. We also looked at 5% spent lime, which is a waste product from lime softening of water, mixed in with 20% leaf compost. And then 5% iron mixed in with 20% leaf compost. And we chose 20% leaf compost because that was kind of the standard, the go-to mix in Minnesota when this project began in 2019. And again, that came from our technical advisory team of stormwater industry professionals. So this was our initial set. We did 14 simulated events in 2019 in the first year as part of phase 1. We were able to conduct 8 simulated events in 2020, despite COVID and all the restrictions and being able to get back on site to do those experiments. So we got a lot of very good data. The first objective for filtration rate didn't show us a lot of differences between these different media mixes. In general, roughly 50% of our volume comes out in the first 5 to 15 minutes. And you can see the graph on the right of this slide. The blue line here is our influent buckets. So we fill our influent buckets with just over 20 liters or kilograms of water. Then we open a valve to let that drain out quickly to simulate a runoff event where you have a rainstorm and it very quickly accumulates in the streets and the roadways, curb and gutter, and then enters into your rain garden quickly. Then the red line here is actually outflow from the bottom of our mesocosms after it's traveled through our different media mixes. And you can see the response is very quick. 50% of the volume within 5 to 15 minutes, 70 to 80% of the volume within about 20 minutes. And most of our mixes had very similar flow characteristics. Actually, almost none had any statistical significance. Only one was slightly faster, and we'll see that towards the end. But we can see we're getting a good filtration rate. Basically, all these media mixes would say what I'd say would pass this filtration rate test. Our second objective was biological. How does it support vegetation growth? And so from the 2019 season, we're actually measuring two things related to vegetation growth. The first is the maximum height. And so this is just max height of the tallest piece of vegetation in each of the mesocosms. And then we take an average of our three different replicates for each media. In 2019, the highest or tallest vegetation was the spent lime with 20% leaf compost mix. We have some others here, but what's interesting, what I saw when I first looked at this plot, is there's actually kind of a natural breakpoint between what is high vegetation growth and what I would consider low vegetation growth. And that was just naturally occurring. It's very interesting. And so the ones that fall into the high category are, as I mentioned, the spent lime with leaf compost, the 10% leaf compost by itself, the 20% food residue compost by itself, the 20% leaf compost, and the 15% biochar plus 20% leaf. Those all generally had higher vegetation or taller vegetation than all the other mixes. So the other mixes in the low category are the 10% food residue, the 20% sphagnum, the 20% reed sedge peat, and the mix of iron with leaf compost. And then of course, our 100% sand doesn't have any organic content, so we didn't expect it to grow anything. But we were surprised that these other mixes in this low category were as low as they were. But this is just vegetation height. We also measured biomass, and we measured the above ground biomass roughly around the time of senescence. So we did that in 2019 and 2020. We'll do that again here in 2021 in about a month or so. That's when we start to see senescence of our switchgrass, which is what we have growing in our mesocosms. We have a monoculture of just switchgrass to try to simplify our experiments. But what's interesting about the biomass here in this graph, I want to explain in specifically because on the vertical axis, we have biomass in grams, again, above ground biomass only, but the scale here is logarithmic. Okay, so just keep that in mind. We had a very large range, multiple orders of magnitude in biomass measurements between our different mesocosms. So we have to plot this on a log scale to be able to see all the data, but just keep that in mind. This is increasing by orders of magnitude or factors of 10. And then along the horizontal axis here, we have our different media mixes. Now, each of these is a pair. So for our 10% food, we have our 2019 bar graph, box and whisker. On the left, we have our 2020 biomass results on the right for 10% food. 20% food is exactly the same. 2019 data on the left, 2020 data on the right. Also, if you can tell in the shading, the 2019 is a little bit darker than the 2020 data. Now, what's interesting with this plot, and again, we're not going to get into all the details of all the data, but just the big trends. I also see kind of a natural breakpoint here for what I would consider high biomass and low biomass. And the number is not important. The 1 gram is not a breakpoint specifically, but just the fact that we have groupings. And what I see for high biomass ratings, I see the 20% food, the 10% leaf, the 20% leaf, the spent lime, and the biochar mixes. They generally have higher biomass than the other mixes that we see, what I would consider the low category. The 10% food, the iron, the sphagnum, the reed sedge, and of course our 100% sand mix. Now, if you remember from our previous slide for maximum height, these are correlating very well. The ones we see with high biomass also had the tallest vegetation. The ones with low biomass generally had the shortest or the lowest vegetation, which makes sense. But that corroborates our metrics for measuring vegetation growth. An interesting surprise that we saw with this data, most of these mixes had fairly consistent vegetation growth from year 1 to year 2. These were brand new mixes in year 1, freshly mixed, freshly seeded, okay? Year 1, we expected lesser vegetation than we would see in year 2 and year 3. We see a little bit of an increase in some of these mixes, but biochar was very surprising. It was right at the border of low, and I'd almost consider it low in year 1, but in year 2, it was the highest biomass of all the mixes. So a very significant change between year 1 and year 2 for the biochar mix, We still don't understand exactly why, but we're excited to see what this turns out to be in year 3. So then our third objective was phosphate release. And this is a big ticket item. We want to limit how much phosphorus is coming out of this system and getting back into our storm sewer, which ultimately goes to surface waters. Now, this is a very complex graph. I want to break it down into parts so we're all looking at the same things. So if you follow my cursor over here on the vertical axis on the left, we have phosphate concentration. And this is in micrograms per liter, which is parts per billion. So these look like big numbers, okay? On the horizontal axis here, we have our different events. As I mentioned, in 2019, we did 14 events, and those are all grouped into media. So that's what I have along the top here. So all of these light blue bars with their error bars for all 14 events are all 10% food compost. The next one is 20% food compost. And so through the year, within a box is the change over those events. And then left to right, we see our different medium mixes. On the far right side of this graph, we have our tank supply. Again, this is synthetic runoff. We mix it. We use river water as our base. We actually spike it with phosphate because in the river, we only have on average 10 to maybe 40 micrograms per liter of phosphate in the water in the river where we collect it at St. Anthony Falls Lab. So we try to spike that up. And that's what this line, this dash line, which I'm going to highlight in red, is the average influent concentration for this first year, which is right around 300 to 400 micrograms per liter. Now, what's important about this red line is all of the bars here are plotting the outflow concentration of phosphate. So as I've indicated on the right side here, if the values of the bars are above the red line, we are exporting phosphorus. We are releasing more phosphorus than what came into the system. If our bars are below the red line, we are removing phosphate. We are capturing phosphate, reducing it from the inflow concentration down to a lower concentration. And what's very interesting is we've also grouped these media into groups of the ones that are releasing phosphate, which include the 10% food, 20% food, 10% leaf, 20% leaf, the spent lime mix, which again is 5% spent lime with 20% leaf compost, the biochar, which is 15% biochar with 20% leaf compost. These are all increasing the phosphate concentration above what's coming into the biofiltration practice, okay? These are the things that we're trying to avoid. And this category in the green box are the ones that are actually capturing phosphate or reducing that concentration as it goes through the media. And that includes the 5% iron that's mixed with 20% leaf compost, the sphagnum peat, which is 20% sphagnum by volume mixed with 80% sand. There's no compost in that mix. It's peat instead of compost. Similar to reed sedge peat is 20% reed sedge peat instead of or in place of 20% leaf compost. And then of course our 100% sand compost. That is, or I'm sorry, sand compost, 100% sand mix. There is no compost in our 100% sand mix. And so as we can see, we have minimal capture in our sand, but we've seen that in previous studies that sand, just because it's silica based, has some calcium in it. It does have some initial kind of absorption or precipitation of phosphate when it's first installed, but that quickly becomes exhausted, as we're kind of seeing in the trend here in the phosphate data. When we look at season 2, 2020, we see very similar trends. And again, exactly the same graph. On the far right side, we have the tank, which is our blue influent. I have our red line, which is our average inflow concentration. In this case, our release are the same media mixes. Our food and leaf-based composts, our spent lime, and our biochar. The ones that capture phosphate are the iron mixes, the 2 peat mixes, and then of course our 100% sand actually is still capturing a small amount of phosphate. So this is very important to understand what our medias can do. And it's very enlightening for how much phosphate can actually come out of these systems just in the first 2 years of operation. So the key takeaways, if we kind of put together for that phase 1, those first 2 seasons, what we've learned is that there are media mixes that will get more vegetation growth. The ones with compost, whether it's leaf compost or food residue compost, and the ones that have spent lime and biochar mixed with compost. However, the downside of the consequences, those are the ones that also release phosphorus. Okay, they're increasing that phosphate concentration. So it's a trade-off. You get more vegetation, but you're releasing phosphorus. So you could be affecting valve balloons downstream. Conversely, the mixes that had iron with 20% compost could capture phosphate, but had less vegetation growth. Similarly, the peat mixes, where we take peat and replace compost with peat, we had less vegetation growth, but we had capture of phosphate. So there is no perfect mix out of these first ones. And if you want to see all the details and all the data and read about the methods, I have the link here for the full report. It's online. I'm happy to share it. If I remember at the end of the presentation, I'll try to drop this into the chat for everyone. Otherwise, feel free to email me and I can send you a direct link or a copy of the final report for phase 1. But we had phase 2, and we're so thankful. I'll talk about funding at the very end. We're so thankful that we were funded to expand this experiment into phase 2. To do that, we took out half of our media mixes. We felt we learned most of what we could learn out of half of these mixes, and we replaced those with new mixes. Again, getting input from our technical advisory team of stormwater professionals. Many of you are probably in the audience today, so thank you. In phase 2, we put in new mixes, and we have so far in 2021 already 8 events. We did a new mix of clean wash sand because we needed a new baseline. We couldn't use our 2-year-old or now almost 3-year-old sand as a comparison for our new mixes. So we just had to put in a new control. We have 10% leaf compost as our baseline now. Our new baseline is going to be 10% leaf compost to see if we drop down that baseline organic material, how does that affect our overall growth and phosphate release? We also added a layered mix where we have 10% leaf compost and sand in the top half of the media and 5% iron and clean sand, no compost, in the bottom half layer. So now we have a layered mix. We also thought, well, can we adjust or optimize our spent lime mix? We saw we did get some reduction in phosphate by spent lime, but it still was not enough to capture all the phosphate coming out of compost. So we said, what if we up the spent lime and decrease the compost? And that's where we have a 10% spent lime and a 10% leaf compost mix. Similarly, we saw sphagnum peat could reduce phosphate, but it couldn't grow plants. So what if we use a peat and compost mix? So now we have 10% sphagnum peat with 10% leaf compost. For vegetation growth, we are finding that we are starting to find a happy medium or a Goldilocks kind of mix. We're starting to optimize these media. So in year 2021, again, this is all preliminary data. A lot of this has been very minimally QA/QC'd, so take it all with a grain of salt. But we still see our original mix. So our original mixes that we kept, the 5 mixes we kept, are the dark solid lines. Okay, so the 5% spent lime, that's an original mix. That's year 3 of operation. Our 15% biochar, solid line, original mix. And that's year 3 now. That's also having good, healthy, tall vegetation. But then our 4 out of our 5 new mixes, these are the dashed lines, the new sphagnum with 10% sphagnum with 10% compost, our leaf mix at 10%, our layered system with iron and compost, and our 10% lime with 10% compost are all getting relatively high or at least middle ground vegetation growth so far this year. And that's very encouraging. It means we're improving some of the medias that had low vegetation growth in the first 2 years with new media that now, even in the first year, are getting us taller vegetation. Some of our legacy mixes, again, our 20% sphagnum didn't really grow a lot of vegetation in the first 2 years. It is growing a bit more this year, but it's still fairly limited. And on our 20% leaf compost from the original mixes, some of those mesocosms really didn't grow a lot, and we're really interested in why. We don't have a good explanation, but for whatever reason, some of the height data for the leaf compost is just not what we expect it to be. And then obviously the sand mixes don't grow hardly anything, which is what we'd expect. It is encouraging that our new sand matches our old sand pretty well, but we still had to do it. We don't have biomass yet for this year, obviously, because we'll do that in about a month. So this winter, we will have that data to share. Preliminary data for phosphate is very interesting. So I'm going to start just with our original mixes. Again, this is year 3 For these mixes, we had our 20% leaf that we kept. We have our 5% spent lime, our 15% biochar, and then our 20% sphagnum, which replaces 20% compost. We see very similar trends to what we saw in the first 2 years. We have mixes that are releasing phosphate. We have the same mixes that are capturing phosphate. So the trend is continuing even after multiple years of service. Now I'm going to switch over to our new mixes, and I'm going to take away the old mixes just so we keep it simple. In our new mixes, we also see release from a number of mixes. Our new 10% leaf compost, which we expected to release phosphate, no surprise, they're releasing phosphorus. Our 10% lime and our 10% leaf is actually releasing phosphorus. We thought we were going to try to get to a middle ground, a compromise between spent lime and leaf. We aren't there yet. It's still increasing the amount of phosphorus that's coming out of this system compared to what comes in. But a big surprise is our 10% sphagnum with 10% leaf mix. Again, we thought this would be kind of a middle ground. We kind of meet in the middle between the high amount of phosphate released by 10% leaf compost and the capture that we saw in the sphagnum in the first 2 years. We thought in the middle. But what we've seen so far, actually higher release initially, although it is trending downward quickly to being less than the 10% leaf now. Initially, it had a lot of phosphate coming out. No surprise, our layer mix is capturing phosphate, but it's nice that in the previous slide we saw it can also grow plants. So that is encouraging. If I put this all together, we can kind of see how things compare. Again, it's not really apples to apples because the original mixes now are in year 3 of operation. The new mixes are all brand new. They're in year 1. So it's not really a fair comparison. But if you want to kind of see how things are shaking out, this is an overall graph for all the data. And again, I can provide this if you're interested, and there will be a recording of this presentation so you can go back and pause here and take a look and see all the details. But the big takeaways that we've learned so far from this phase 2 preliminary is that our best plant growers are 5% spent lime and 20% leaf. That's an original mix, 3 years old now. The biochar with leaf compost, again, another original mix, is one of our big plant growers. But our new mix of sphagnum and leaf compost is also producing a lot of vegetation so far this year, which is very encouraging. Our leaf compost at 10%, another new mix, is producing a lot of growth, as well as our 10% leaf layered over 5% iron. That is where we're trying to meet middle ground. And I feel like that one is the best in terms of trying to find a happy medium between plant growth and phosphate, because it captures phosphate, but it still does grow plants. And then, of course, one of our other new mixes, our spent lime and leaf compost, is also doing very well so far this year in year 1. Phosphate capturers is a much shorter list. Obviously, the original mix of 5% iron and compost is capturing a lot of phosphate. Our new mix of leaf compost layered over 5% iron is also capturing a lot of phosphate. And then our 2 original sphagnum and reed sedge peat mixes also captured a lot of phosphate. And sphagnum continues to in year 3. But what are the surprises? Well, the vegetation growth change in biochar. After year 1, in year 1, it was very minimal. In year 2 and year 3, it is explosive compared to year 1 at least. So that is very surprising. We're still trying to understand what's going on there. We're very surprised by that phosphate release of 10% sphagnum and 10% leaf compost. We're not sure why that is. We expected it to be less than 10% leaf. And it is now after 8 events, but initially it was much higher. So that's a really big surprise and really unknown for us at this point. And then, of course, we were a bit surprised that our happy medium of spent lime, trying to meet in the middle. We started at 5% spent lime and 20% leaf. We tried to go to 10% spent lime and 10% leaf to kind of meet in the middle. So far, they're about the same. So we're not sure we really optimized or improved there at all. But we do have more work to do. We have another field season next year. And we do, obviously, this is all preliminary data. We do need to go through the full QA/QC and finish out this field season. As I mentioned at the top, this winter, we hope to do our low organic content germination tests, seed germination tests, to find out how low can you go. Maybe 10% is kind of the low end. We don't know yet. Next spring, we're going to introduce road salts to see if that impacts phosphate release and plant growth. We're also hoping in future years, there is a proposal out there that could look at contaminants of emerging concern and how they can be removed by biofiltration media. We're very excited that that is a distinct possibility of future research using this experimental facility. It could lead to really good work. And now that we've narrowed down at least a little bit into an ideal or optimal biofiltration media design, this could then see, can it target other pollutants? And then obviously we're always open to other ideas. We're open to other collaborations and other potential projects. I do want to acknowledge funding before I close up here. This is funded by the Minnesota Stormwater Research and Technology Transfer Program through the University of Minnesota that gets funds from the Clean Water Fund through the Minnesota Clean Water Council and the Land and Legacy Amendment. If you want to find out more information about any of these organizations, links are here. John also shared them as well. I also have to note that everything I've said today is my opinion. It does not reflect any of the funding agencies, but just my opinion as a researcher on this topic. And I also want to acknowledge before I close all the people and organizations that have supported this work. The funding agencies, the Water Resources Center, and the administration to be able to get this work and contracts through. As I mentioned multiple times, we have stormwater professionals and technical experts on a technical advisory team that met twice so far and has withstood many of my emails with questions about how we can help them do better design work. And so we're very thankful for all of that input. We also have a number of organizations that donated materials for this work, as well as a long list of staff and students at St. Anthony Falls Laboratory to help make this work possible. Mix of graduate students, undergraduate students, and staff. I've listed many of them here. I'm sure I've forgotten several. I'm very, very thankful. We could not do this work without all of this help. Before I let you go, I do want to reiterate stormwater updates. This is an email newsletter that comes out a few times a year. If you are not signed up for this, this is how you can get notified about future stormwater research spotlights and stormwater seminar series events, as well as research projects from the University of Minnesota on stormwater, such as this project was highlighted in updates in February of this year. So if you want to get the late breaking news on stormwater research projects that are coming out from the University of Minnesota, I encourage you to sign up for updates at our website here. You can find past issues of updates. You can also find a full list of all of our publications and reports. So with that, after going long, I'm going to thank you for your attention. Thank you for participating and joining us today. We have a number of questions I see in the Q&A. Obviously, you can email me if we run out of time today with any other questions or for more information. With that, I'll turn it back to John Bellata to moderate.
Thank you, Andy, for your great presentation and great work, and to your entire team. We do have quite a few questions. We'll tackle as many as we can here. We'll probably end by 11:10, just so you all know. And so you also know that Andy's presentation is recorded, and we will post that online, as well as give him and the team a chance to answer these questions in more detail, and we'll send that out as well. But let's tackle some of these questions. And so the first up here, Andy, is from what a participant could tell, you are only relying on media to capture the phosphorus coming through the system. Are you also looking into the types of plants that may use the most phosphorus, thus remove it from the system through biomass growth and potentially permanently remove through mowing, raking, baling, taking that biomass off-site?
Yeah, that's a very, very good question. We needed to keep our experiment somewhat controlled in the number of variables because we're actually measuring quite a bit here. So we did a monoculture of switchgrass in our mesocosms. So there is no plant diversity. We're not looking at other plants. We do weeds and growth on some things, and that gets taken care of in our biomass at the end that's separated from our switchgrass. But we are not quantifying how much phosphorus is being taken up or transformed by the plants or the soil biology, okay? However, that is occurring in these mesocosms. So even though we are really focusing on the media mixes, whether it's iron or spent lime or biochar or compost and peat, really all of those other factors, all of those other mechanisms, because they are outdoor experiments, are also occurring. We are lumping all of that together into the overall performance of the system, with the thought that if the media can support healthy vegetation and healthy microbial communities, you will get all of those benefits. Now, in the low organic content seed germination test that I mentioned very briefly, again, that is just looking at seed germination. Can we get seeds to germinate quickly and abundantly in these different media mixes? And that will look at 10 different species, and it's a wide range of species. Again, we're not going to quantify or look at uptake. We're not going to look at microbiology or transformations or any of that. We just want to look at seed germination with the assumption that if we have healthy, diverse vegetation, we will benefit from all of those processes. So thank you for that question. I will also mention, there is some very good literature out there. Bill Lucas did a really good study, mesocosm experiment, looking at vegetation specifically and separating out nitrogen and phosphate and what was taken up by the plants or transformed by the plants and the rhizosphere and what was conducted by the media itself. So Bill Lucas, Lucas and Greenway are a number of publications on that work. There's a number of others, but I'll just mention that and I can be in touch if you want more.
Great, thanks, Andy. Since these experiments and the mesocolumns are going from year to year, the next question is, are you taking into account the nutrient buildup and reintroduced to the system from the previous growth of biomass?
Very good question. Very good question. So what's possibly unique or a little bit different about the way we're running our experiments compared to a typical rain garden or biofiltration practice? We are using river water as our source. So there is some organic material. There's algae and things in the water, but it's not nearly as much organic matter as I would expect in runoff. So one, it's not getting the same amount of organic matter in with inflow as a typical rain garden or biofiltration practice would get. Two, the vegetation that we grow in the mesocosms, we actually harvest. We do this above ground biomass, and we literally collect everything above the surface to quantify biomass. So if you wanted to translate that to the field, it'd be like going out and literally cutting everything down right to the ground, collecting it all and taking it out. So unlike a natural rain garden or bioretention, which maybe might not even get cut back, right? The natural senescence or the dying off of the vegetation literally stays right in the rain garden. That nutrient cycle is more consistent and carry over from year to year, whereas in our experiments, a lot of that biomass, especially all that above ground biomass, is taken out of the system. The below ground biomass, all the root structures and all that is remaining intact. But that is a big difference between what we are doing and what you would see in the field. And that's just kind of a nature of being able to measure the metrics that we're using.
Thanks, Andy. One more question on nutrients, and then I'm going to try to group some of these questions on media mixes. But first, you're spiking your samples with bioavailable phosphorus, but in reality, practices capture particulate-bound phosphorus as well. Are you looking at that? Are you planning to take a look at that?
Good, another good question. So as I just mentioned in the previous answer, we're using river water as our base. And you're exactly correct. We are spiking this with soluble phosphorus specifically. And so we're increasing the soluble fraction the soluble concentration from anywhere from 10 to 40 micrograms per liter background in the river to our target is about 300 micrograms per liter in the tank that we add to our buckets that we then spike into our mesocosms. And we intentionally are not adding sediment to the system. Again, in the river, you can see in the photo here, we're literally on a pretty wide section of the Mississippi River where we take our water from. There's not a lot of sediment. There's some very fine suspended sediment. And as I mentioned, there's algae in there. But otherwise, the particulate load is very minimal. In addition to that, we fill our tank, our supply tank. We have a 500-gallon supply tank. We fill that the day before. And so actually, the water has anywhere from 16 hours or so to settle out some of that sediment that's in the water as well. So we are intentionally not incorporating sediment because we did not want clogging at the surface due to sediment to affect the filtration rate that's really controlled by the media. That's what we're trying to measure. We're measuring different media mixes, and we wanted to know, is there a difference between the media for filtration rate? And we didn't want to get that confused with clogging and limits of filtration rate. So we're intentionally not including sediment for the clogging rate, but then in addition to that, then we also are not measuring particulate phosphorus. However, because they're filter-based systems, we would expect 80% or so of the particulate phase of phosphorus to be captured. if that gets remobilized, if that desorbs from the particles, or if it breaks down because it's organic matter and goes into the system. Again, the media, we're trying to find a media that can capture phosphate no matter how much is coming into the system.
Well, great, Andy. So we have a number of questions here about media mixes. I don't have all your reports memorized, but I assume the team talks more about the specifications of each of the mixes in the studies.
Yeah.
But questions here, do you have specifications, various compost mixes? Another question is, what kind of sand were you using? Let's start with those two.
Yeah, those are good questions. So again, the very common specified base sand in almost all of these, especially sand filters, but also bioretention, biofiltration practices, is an ASTM C33 concrete sand. And it's the same clean wash sand that you can go and buy at any hardware store in a big bag to make concrete. It's very common. You can get it from any aggregate supplier. And it should be clean washed sand. That's kind of critical for stormwater treatment. That's the base for sand. The compost, the biochar, the spent lime, the iron, all of that, all the details for where that came from and what details we have on that material is all in the final report from phase 1. It'll also be detailed in the final report for phase 2. If you want that information, just contact me and let me know. On my previous slide, one of the suppliers, we went with Plasteads. He's a local supplier for many of these media mixes. They do an automatic blending for rain garden mix and a lot of other media mixes. We wanted something that was easily accessible to anybody. And I know a large number of contractors use Plasteads here locally in the Minneapolis, St. Paul, Twin Cities metro area. And so we wanted to get what contractors could get. So we just use that, and they provide all the materials, and we have details for that, and I can share that if you're interested.
Great, thanks, Andy. Another question here. You might have talked about this one, but are the additives mixed into the sand for homogeneous? Were they layered on top? What was your approach on that?
Great, that's a good question too, and that actually follows on very well with the previous question. As I mentioned, we got all our materials from Plasteads. We asked them to blend it for us because, again, if any of you working with contractors and working on sites and projects, sometimes you don't even know how they mix the media, right? Do they get it all delivered and poured on the parking lot and stirred up with a couple of front-end loaders or something? We didn't want to deal or incorporate that level of variability in our experiments. So we asked Plasteads to pre-mix all of our media. Everything I talked about, with the exception of the layered mix, everything else I talked about, it is fully mixed, top to bottom, 18 inches of media, okay? So if it's leaf compost and sand, it's the full mix, top to bottom throughout. If it's spent lime and compost and sand, fully mixed, top to bottom, all three of them. The only exception is the layered mix. That's new for this year. What we did differently there is we did sand and compost fully mixed in the top 9 inches, and then iron and sand fully mixed in the bottom 9 inches. So that's the only media mix where there's a separation from top to bottom. And that's because we have a layered sand and compost on top, layered iron and sand on the bottom.
Well, thank you, Andy. Again, for all our participants, we'll go till about 11:10. We'll see if we can get through a few more of these questions. Again, we'll send all these questions out to you following the seminar. I want to change gears a little bit here. I see a couple questions regarding pH. And so one of those was in the chat and one is in the Q&A panel, but I think they're both really good. I can't find the one in the chat, but I think it had related to, do you test pH of the incoming water and the river water, and how is that related to your methodology? And then related also on pH was thinking about the pH of the sphagnum peat moss, or maybe any other media mixes. How is that related to your work and taken into account?
Yeah, so we are measuring pH. We do have background pH for the river that we're measuring, but as we fill the tank before we add the phosphate, we are measuring the pH of all the water that comes out, of all isocosms. There is a slight elevation in pH from the river to post-treatment, okay? But it's a very slight elevation. I want to say The outflow, I don't know if I can remember the river off the top of my head, but I want to say the outflow from all the mesocosms is in the range of 8 to 9 for pH. So it is elevated a bit higher than you might expect. But again, a lot of these systems are a mix, right? We have a lot of sand in there, and some of the fines in the sand could be affecting pH. There could be other things in the media that affects pH. We were very interested in the spent lime, because spent lime, I didn't go into this, but it's essentially lime, right? It's calcium carbonate that's used for lime softening. That can have a big impact on pH. It has a lot of alkalinity there with the carbonate species. So we thought that could have a big impact on pH. It was a significant difference, statistically significant difference, but it was pretty small. It's not like the pH was up at 10 or 11 or anything like that. It was slightly elevated above the rest. But we are measuring pH. Interestingly, with the sphagnum and the reed sedge peat, again, with a lot of humic soils and things like that, you might expect the pH to drop pretty considerably. A lot of wetlands have pretty low pH. A lot of humic soils have pretty low pH, soil pH. We didn't see that in this. And we don't know if that's because the sphagnum and the reed sedge is just a very mature material. It's stable. It doesn't have a lot of impacts on the water as it's going through, or a combination with the very short residence time. Again, 50% of the water out in less than 15 minutes and 70-80% of the water out in the first 20 or 30 minutes. So the water is getting through very quickly. Low contact time, fairly stable materials. We're not having a big impact on pH. I have all that data. I can share that though, if you want to see it.
Thanks, Andy. A lot of participants today are really interested in the sand mixes here, I see. Yeah. So we're going to go back to a couple sand mix questions. I'm going to try to combine these. Mm-hmm. So thanks, Michelle, for asking this question. But is manufactured sand acceptable, or do you insist on natural sand? The manufacturer's crushed stuff is angular, and in Maryland, we don't like it because it tends to knit together and decreases the flow rates. So let's start with that one, and then we have another sand media question.
Yeah, that's a good question. It's something we haven't considered. I certainly haven't measured it. We haven't looked at a crushed or an angular sand versus a more round kind of natural sand. I do know that there is a mix. In Minnesota, we do have a lot of quarried sand. So some of that is going to be angular just because of the nature of the quarrying process. But I don't know if I can say what the difference is. We certainly haven't measured it. I haven't read any literature on that. So I don't have a good answer on that. It's a very interesting question though. And it is something that I want to look into now that you've asked it. So that's about as best as I can do.
Great. One last question here on sand, and then I'll conclude with a final question for you, Andy. In trying to optimize filtration rate and plant growth, in addition to phosphorus export, why only use relatively uniform clean sand as the base for all the mixes versus some natural sand? Again, related to the same question we just addressed.
Yeah, that's we actually see. In a lot of stormwater manuals around the country, there are mixes, and even in the Minnesota Stormwater Manual, there are mixes that allow for topsoil or native soils. I really like to see when they have that there's a caveat for how much clay and fines are included, because I have seen mixes that don't specify an upper limit on clay and fines, and then those medias can clog really quickly if your native soil is just a lot of fines in them. However, why we chose what we chose to mix. We were trying to see with just compost, to keep it simple, compost, and then a few of these amendments, spent lime, biochar, iron, and then the peats. Is there a measurable difference there? We didn't know if there would be. We actually expected, with spent lime being what it is, it's actually a very fine powdery cake material. We actually expected that to be very mobile and very fine and possibly affect the filtration rates. We didn't see that in our data, but again, it's relatively small amounts, 5%, 10%. So we haven't seen that yet. But that was the main reason, is I wanted everything to be relatively consistent and comparison apples to apples. And we wanted to see if just having these initial things affect filtration rate, then now in our next steps, we could look at incorporating fines, we could incorporate topsoil, we could incorporate native soils to look at some of those other impacts. The challenge there is the variability is immense, right? Topsoil here, topsoil there is very different in a very small scale. Native soils here and there is very different on a very small scale. So we would have to quantify that very well. And would that relate to everybody? Maybe not. Maybe if we do just a particle size distribution, a hydrometer test for the fines, we characterize it that way, and then organic content, we characterize it that way. Maybe that's good. But that's just right now, it's kind of beyond the scope of what we wanted to do with this so far, but certainly something we could do in the future.
Well, great. Well, thank you, Andy, and thank you everyone for submitting a lot of great questions. Again, we'll send these over to Andy to follow up on. And so I want to give a virtual round of applause to Andy for his presentation and your entire team for all the great work that you've done. And so we will start to wrap up our seminar here for today. And so with that, I guess I would like to encourage you all to rejoin us in October, November, and December as we continue our Minnesota Stormwater Research Spotlights and the Stormwater Seminar Series. A reminder that, again, that all of our presentations are recorded and posted, so you may go back to those and view them in the future. So thank you again, Andy, the entire team, the team over at SAFL, and thank you all for giving us an hour today to learn about this great work, and join us again in the future. Have yourselves a fantastic day.